SwayamPrabha. Digital India. Educated India.
Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute, Pusa, New Delhi. This lecture pertains to the course Agronomy of Oilseed, Fiber and Sugar Crops. The title of this lecture is Nutrient, Water, Weed, Insect Pest and Disease Management in Castor and Processing of its Produce, means what happens after harvest of the crop. So, learning objectives are to achieve an overall understanding of the nutrient, water and weed management practices in castor, to detail the insect pest and disease management and post-harvest handling of castor produce. Let us see some difficult words. Desiccant, any substance or mixture of substances used to accelerate the drying of plant tissues. Dicotyledon, a member of the dicotyledony, one of two classes of angiosperms usually characterized by two seed leaves. These are called cotyledons with net venation. Mode of action, the mechanism by which pesticides affect target organisms. Native plant, a plant species indigenous to an area, not introduced from another environment or area. Nematode, unsegmented worm with elongated rounded body, pointed at both ends, piercing mouth parts, mostly free living but parasitic also. Plant disease triangle, diagrammatic representation of three key factors contributing to plant diseases. This is host, pathogen and environment. Race or strain, organisms of same species and variety differing in ability to parasitize varieties of a given host or that differ in their reaction to pesticides, called race or strain. Root, the part of a plant usually below ground that holds the plant in position, draws water and nutrients from the soil and stores the food. Shoot, the aerial or part of a plant, a stem and all of its dependent parts. In general, all the above ground parts of a plant are called as shoot. Side dress, to apply a fertilizer, pesticide or soil amendment to one side of a growing plant or rows of plants, either by surface application or injection into the soil. Sodic soil, a soil with pH more than 8.5 that contains sufficient sodium to adversely affect plant growth, plant production and soil structure. Now we go to the nutrient management, which is also important part, and as many of the students understand that nutrient management includes the choice of right kind of fertilizer or right source of the nutrient, right method of application, right time of application and right dose or amount of application. So scheduling of nutrient management is very, very important. So one of the most effective means of increasing the yield of castor is through fertilizer or by providing nutrients. Even under dryland conditions, the use of fertilizer has resulted in significant increases in seed yield of castor. Fertilizers help in development of deeper root system to extract moisture from deep layer. Particularly, there is effect of phosphorus fertilization more on the growth of the roots. However, so far castor is found to respond to only major nutrients, more particularly to nitrogen and phosphorus. In most of the fertilizer trials with castor varieties, about 45 kg nitrogen and 60 kg phosphorus per hectare were optimal, and higher doses of fertilizers resulted in increased yields of castor only during years of rainfall or under irrigated conditions. As the castor crop is rain-fed crop and it does not respond much to the fertilizers. Therefore, it requires less quantity of nutrients like 40 kg nitrogen and 60 kg phosphorus per hectare. But if irrigation is provided, if rainfall is more, then in that case we can increase the dose of the fertilizers. Application of fertilizers may reduce oil content in seeds, but total oil yield is higher due to increased seed yields with the use of fertilizer. So overall, you can see percentage oil in seed can decrease, but total oil yield will increase due to application of fertilizers. Manures, particularly farmyard manure, have a positive impact on yield of castor. Application of 20 tons per hectare of manure gave 0.1 to 0.15 ton per hectare higher castor yields, which was equivalent to the effect of application of a fertilizer dose of nitrogen 45 and phosphorus 60. So organic manures also have a better choice to supply nutrients. There are genotypic differences in the degree of response as per the yield potential for fertilizer application. In general, hybrids respond better compared to varieties. The response to fertilization is modified by the associated moisture conditions and pests and diseases. So definitely there is interaction of nutrients or interaction of fertilizer and kind of plant or varieties. In general, your hybrids respond better or higher than the varieties, and also the response is moisture dependent as well. As in case of rain-fed conditions, you need less fertilizer. Under irrigated condition, one can increase the doses of the fertilizers. Higher fertilization to varieties with low yield potential causes excessive vegetative growth and delay in maturity. So therefore, there may not be a balance in the vegetative and reproductive growth. A variety which is not responsive to nutrients, if you add more nutrients to that variety, it will attain more vegetative growth and there will be less emphasis on the reproductive growth. So eventually the seed yield will be decreased. The height of plant is also affected by fertilizer applications, and a variation to the extent of 0.5 to 1.25 meter was noticed between varieties of phosphate application. The castor crop responds profitably to nitrogen and phosphorus fertilizers, and the response to K fertilizer is generally not noticed, and the soil reserves of K is sufficient for castor. It is always advisable to apply nutrients to castor based on the status of nutrient availability from the soil, both for major and micronutrients. It is a better idea to get our soils tested, or soil test based application of nutrient is advisable. Whatever recommendations comes from the soil test, that should be applied. The general fertilizer recommendations are for rain-fed conditions 40-40-0. This is kg nitrogen phosphorus in form of P2O5, K2O per hectare. And in this case, nitrogen is applied in splits 50% basal and 50% at 30 to 35 days after sowing, and half quantity the phosphorus dose should be applied as basal. Under irrigated conditions, 40-40-40 kg nitrogen P2O5 and K2O per hectare as basal application, plus 10 kg nitrogen per hectare top dressed at each picking, starting from 90 days after sowing at 30 days interval. Under situations of high input, maximum yield, that is in Gujarat, was obtained when total nitrogen uses would be beyond 150 kilograms per hectare. And here you can also see the yield potential is 5 to 6 tons seed per hectare. If you are harvesting 5 to 6 tons of seed per hectare from the castor crop, we should use at least 150 kg nitrogen per hectare. Though there is no widespread deficiency noticed for secondary and micronutrients, as students understand that secondary nutrients include calcium, magnesium and sulfur, and micronutrients include about 8 nutrients that are zinc, iron, molybdenum, copper, boron, chloride, etc. So rarely the response to these nutrients are found in case of castor. The use of superphosphate as a source of phosphorus can meet the requirement of sulfur and calcium unless they are seriously deficient in certain situations. That means if farmers are continuously using single superphosphate fertilizer, then there is no need for sulfur or calcium application, as by default this fertilizer will supply enough quantity of calcium and sulfur to the soil. Responses to micronutrients like zinc and iron and secondary nutrients like sulfur have been reported widely in India, but only in conditions where these nutrients are deficient. Now see, integrated nutrient management practices are important, and in integrated nutrient management practices we include crop rotations with legumes, green manuring, phosphate solubilizing bacteria will greatly reduce the cost of fertilizers besides improving the soil fertility. As we understand that in integrated nutrient management, we need to combine several sources or all the sources available for the nutrient application like organic manures, fertilizers and biofertilizers. And crop rotations also have very important role in nutrient management if we include leguminous crops in the rotations. Studies at CRIDA, Central Research Institute on Dryland Agriculture, indicated that the cowpea grown in between the rows of castor and incorporated as green manure at the rate of 10 tons per hectare around 45 days after sowing added 30 kg nitrogen per hectare. Alternatively, green leaf manuring through subabule is called as Leucina leucocephala, also contributed to the pool of nitrogen. So we can do in situ green manuring as well as ex situ green manuring. For intercropping systems involving castor, it is advisable to apply recommended doses of nutrients to each crop in proportion to its area occupied in the system. Now, you have seen the nutrient management practices in castor crop. Let us now see the irrigation management practices in castor crop. So, it is highly drought tolerant crop but responds favorably to irrigation. The magnitude of response to irrigation is higher with hybrids than with varieties. That means your hybrids require more water than the varieties. The root growth of castor under irrigated conditions is restricted to surface layer, whereas the root grows deeper under rain-fed situation. So, in rain-fed situation, there is more deeper roots. Mainly in want of water, these roots go deep. Castor is reported to respond very well to drip irrigation system with high productivity, reduced water use and increased profitability. So, these days we are experiencing the shortage of water in all spheres of life, and it also includes agriculture. There are huge shortages. So, what can be done under these systems? We need to follow the efficient irrigation management practices. So, in this case, drip irrigation is an option, and drip, because it is widely planted crop, so drip irrigation can be practiced in this crop, and it will definitely save lot of water and increase the productivity of the crop, and it will also help in decreasing the weed growth in the inter-row spaces. The ideal water management schedule for castor is irrigation schedule during first half of the vegetative period and during period of mass flowering is critical. Retention of moisture in active root zone by irrigation at 70% field capacity during vegetative and ripening periods and at 80% field capacity during the flowering stage. High moisture during maturity causes a spurt of new leaves and vegetative shoots and delayed maturity. So, we need to have the ideal moisture content. We should not have moisture content always at field capacity. Let it be depleted up to 20% or 30%, and then we can apply the irrigation. Growing castor during the winter season would provide optimal conditions for growth and production, and when combined with drip irrigation and fertigation can achieve the highest yields. A marked reduction in seasonal irrigation water needs of castor achieved under drip when compared to the conventional surface method of irrigation. In conventional methods like border strip method or flood methods, the efficiency of water use is less. However, if we follow drip irrigation system, the water productivity or water use efficiency increases. Now after nutrient management and water management, now we see the weed management in castor. So definitely like in any other crop, weed also cause problem in this crop, and they compete for space, they compete for light, they compete for water, they compete for nutrients, or in short we can say that weeds compete with the main crop or castor crop for resources, and finally the resource availability to the crop is reduced, and which results in poor growth of the crop as well as poor seed yield. So crop is highly susceptible to weed competition, particularly in the initial period of growth. So initial growth of castor is slow and crop is widely spaced, which encourages weed growth. Critical period for weed competition is 45 to 50 days after sowing. So you can see during this period there should not be any weed growth in the field. Clean cultivation in terms of summer plowing and thorough seedbed preparation eliminates weed to a large extent before sowing. So it is better to go for clean cultivation and deep plowing. Now see what are the important weed species that infest the castor crop. It may include Echinocloa colonum, Eleuropus villosus, Dactylioctinium aegyptium, Brachyria species, Cyprus rotundus, Commelina bengalensis, Digera arvensis, Indigofera glandulosa, and Amaranthus viridis. So these are some of the important species that are found in the field of castor crop. And you can see or you can identify these weeds with their pictures here. First of all, on the left side you can see the pig weed. It is known as Amaranthus viridis hook F. It belongs to the family Amaranthaceae. Next is swine cress. You can see in this picture how it looks. Swine cress. It is Coronopus didymus L, and family is Brassicaceae. Next weed is black nightshade, Solanum nigrum L, and family is Solanaceae. So you can identify this weed also. Next weed is false amaranth. The scientific name is Digera arvensis. Family is Amaranthaceae. Next in the center you can see rabbit or crowfoot grass, Dactylioctinium aegyptium. And you can see next is crabgrass, Digitaria sanguinalis, and family is grass family, which is Poaceae. The next you can identify it is Bermuda grass, which is Cynodon dactylan or doob grass, and then purple nut sage in the center, called as Cyprus rotundus or Motha. Family is Cyperaceae, and in the right side you can see flat sage, Cyprus iria. This is annual plant, and Cyperaceae family. However, Cyprus rotundus is perennial. So use of a pre-emergence herbicide like fluchlorelin or pendimethalin at the time of sowing in wet soil provides good control of most broad-leaved weeds and few grasses. So fluchlorelin is basically a pre-plant incorporated herbicide, and pendimethalin pre-emergence herbicide. Intercultivation by hoeing and hand weeding once at 30 to 35 days after sowing provides a competition-free environment for slow-growing castor, which later can cover up the land area and tolerate and suppress the weeds. So intercultural operation or hoeing can be done, and these kind of operations can be combined with herbicide application. Several mechanical methods of weed control like the use of brush cutter and intercultivator removes and loosens soil in the interrow, facilitating better rainfall infiltration and soil moisture conservation. Effective post-emergence herbicide have been identified to manage weeds at regular interval in long duration crop. So it is advisable to go for integrated weed management practices. We can combine manual, mechanical and chemical methods of weed control. However, herbicides are available for pre-emergence application as well as post-emergence application in castor crop. So here application of pendimethalin 1 kg per hectare, that is 6.7 ml per liter water as pre-emergence, plus Quizalow FOP ethyl 0.05 kg per hectare. Remember, the dose of Quizalofop is very small, 0.05 kg per hectare, and that is 2 ml per liter post emergence at 25 days after sowing, plus intercultivation followed by hand weeding at 60 days after sowing, resulted in effective weed management of castor crop. Now, after weed management, we know that crop protection is also needed from the insect pests and diseases. So, let us start insect pest management in castor crop. So, no doubt castor crop has fewer insect pests and diseases limiting its production, but still at times some insect pests and diseases can cause damage to the crop. The feature of the crop being hardy applies to its tolerance to biotic stresses, especially defoliator. So, there are certain insects which are called as defoliator. They remove or they cut the leaves. Due to the perennial nature of the indeterminate plant types, there is a strong compensation for covering up the loss of earlier stages. So, it is having kind of indeterminate growth habits, and it can produce branches, it can produce leaves again and again. So, if some is eaten by the insects, then it can go for recovery also. Besides, the castor has a high source size, and defoliation of more than 50% has not caused significant yield losses. Means even if some leaves are eaten by insects, then it is not going to harm the seed yield. Castor plants were able to completely regrow their leaf area when the defoliation occurred during the vegetative stage. So, however, this initial damage reduced the maximum leaf area obtained by the plant. Defoliation treatments did not influence the number of fruits per raceme, but caused a steep reduction in the number of racemes per plant. Any level of defoliation repeated along the plant growth cycle was found to cause a reduction in the oil yield. The waxy coat present on the plant is known to avoid certain insect pests. This is very important defense mechanism developed by the castor plant. So, it has got some waxy coat that prevent damage by many insect pests and diseases. Also, the ricin, you can remember, ricin is a toxin present in the leaf, prevents some insect pests. So, it has its own system, its own defense mechanism to face the insect pests. Now, first insect pest is semi-looper. So, it is A. genata L is major pest occurring throughout the crop growth period. Means this insect can come at any stage of the crop growth. Freshly hatched larvae feed gregariously. Gregariously means they eat in group. Scraping the chlorophyll soon disperse. Sometimes feeding is so heavy that only petioles and branches are left behind. Most of the parts of the leaves are eaten up by the semi-looper. Caterpillars feed on leaves and cause sometimes complete devastation of the crop. If environment is favorable for the insect or caterpillar, then they can do more damage. However, if environment is not favorable, then plant can escape the attack of the semi-looper insect. And you can see the insect on the right side. It is quite distinctive insect, caterpillar, and it is eating on the leaves. Now, you can have more close view of the caterpillar of this insect. Now, hand picking and destroying of larvae in early stages is an option for management, and one can release or preserving natural egg parasites, for example, T. prodeter, Trichogramma chilonense, and larval parasite like M. maculipennis from the second fortnight of August. So, some options are available for biological control of semi-looper. At present severity, spray malathion 50 EC 2 liter per hectare. Means if there is pest severity, we can go for spraying of malathion 50 EC at 2 liter per hectare. And remember, this 2 liter solution should be mixed with 500 liters of water for spraying in 1 hectare area. Next insect is red hairy caterpillar. As the name suggests, it has got red hair. You can see picture on the right side. It has got some black hairs also, but dominating hairs are red in color. So its name is Insecta albistriga. Caterpillars scrape the under surface of the leaf when they are in neonate stage, means very early stage. Later the scraped patches of the leaves look like thin and papery. Fully grown larvae devour the entire foliage, flowers and growing points. So many times it can cause severe damage to the castor crop. How to manage this red hairy caterpillar? Collection and destruction of egg masses and hand picking of larvae. This is one. Number two, erection of light traps soon after the monsoon for 20 to 45 days, collecting and killing of the adult moths. So it is necessary to kill the adult moth to prevent the next generation. The dispersing larvae of hairy caterpillar from one field to another can be checked by digging trenches across the direction of their march and prompt destruction of larvae. So in these trenches one can put some water so that these larvae cannot cross and they will die. Vegetative traps utilizing Jatropha like wild castor or Ipomia prevent the migration of the grown-up larvae. Dusting of quinol phos. Quinol phos is an insecticide and it can be dusted at the rate of 25 to 30 kg per hectare, and it would effectively control the red hairy caterpillar larvae. Spray quinol phos or dimethoate 2 ml per liter of water at the rate of 500 liter solution per hectare. Now next insect is slug caterpillar and it is Parasa lapida. Larva feeds on larvae voraciously, leaving only midrib and veins, resulting in severe defoliation. So they eats mainly on the leaves. You can see greenish color of the larvae on the right side. Picking gregarious caterpillars means they come in groups and cocoons and destroying them in water with a film of kerosene. This is another option to control slug caterpillar. Spray chlorpyrifos at the rate of 2 ml per liter, 500 liters per hectare solution. Next insect is tobacco caterpillar. You can see the damaging stage, which is larvae or caterpillar on the right side. So Spodoptera litura. The early instar larvae feed gregariously on the underside of the leaf, leading to skeletonization of the leaves. Means they will eat all the green parts of the leaves and only the skeleton, which is veins, may be seen. Sometimes the feeding is so heavy that only petioles and branches are left behind due to tobacco caterpillar. So collect and destroy egg masses and caterpillars and plow fields so as to expose pupae to predators and parasitoids. This is one option. Next option is monitoring pests with pheromone traps. 4 to 5 per acre helps in timely treatment. To control early stage larvae, spray neem seed kernel extract at the rate of 5% or chlorpyrifos 2.5 ml per liter or quinol phos 2 ml per liter of water or neem oil 5 ml per liter of water. So number of options are available. One need not to use all these. Only one of them is to be chosen and applied or sprayed to control tobacco caterpillar. Next important insect is Bihar hairy caterpillar. So its scientific name is Spilosoma obliqua. Young larvae feed gregariously, mostly on under surface of leaves. A caterpillar of later stages defoliates the field completely. Collection and destruction of eggs, this is number one option to control, and early stage larvae. Spray neem seed kernel extract NSKE, that is 5% to kill early stage larvae. For grown up larvae, spray quinol phos at the rate of chlorpyrifos at the rate 2 ml per liter of water and solution amount is 500 liter. So you can see that the treatment or control varies with the stage of the larvae. Shoot and capsule borer. So again, damage is done by caterpillar. On the right side, you can see the pinkish caterpillar and damage being done to the crop. So it is caterpillars bore into the shoot and seed capsules and cause extensive damage by this shoot and capsule borer. A webbing of capsules along with excreta is seen. Damaged capsules webbed together, peduncle and capsules having galleries made of silk and frass. The infested shoots and seed capsules may be collected and destroyed, and spray any of the following insecticides thrice from flowering at 3 weeks interval. This is important. One can take dimethoate 2 ml per liter or quinolphos 2 ml per liter can be sprayed at the rate of 500 liter solution per hectare. Next, number 7 insect is white fly, and it is T. ricini scientific name, and in this case both nymph and adults suck the sap from the leaves. Leaves appear sickly and sooty mold is developed. Sooty means blackish color, mold will also come. And water soaked spots on leaves and become yellow and dry up. Stunted plant growth and shedding of fruit bodies. So sometimes it can be very damaging insect. White flies can be effectively attracted and controlled by yellow sticky traps. Some traps can be prepared which are coated with grease or sticky oily materials. Spray trizophos is an insecticide, 2.5 ml per liter of water, or acephate 2 ml per liter of water, and again solution will be 500 liters per hectare. Or in place of trizophos, one can go for spraying of 5% neem seed kernel extract with any sticky material. And use of synthetic pyrethroids increases the intensity of whitefly. So we should not use synthetic pyrethroids. Better to use trizophos or neem seed kernel extract to control the white flies. Now we go to the castor diseases and their management. Here we will discuss the important one, which are more important diseases. So first one is seedling blight. It is caused by Phytophthora colocacea, and it can be at different stages. So first form is dead seedling. The disease initially makes it look on each surface of cotyledonary leaves as round patch of dull green color. The color is dull green that shortly spreads to the point of attachment, inflicting or affecting leaf to rot and droop down. The drooping of the leaves will be there. Drooping of the leaves means hanging. The leaves will hang from the branches. So, and you can see on the right side, it is almost death of the seedling, initial stages. Next is spot on older leaf. So infection additionally spreads to stem with the result that the seed plant is killed either because of the destruction of growing point or by the collapse of the stem. So on the right side you can see the symptoms on the leaves. It is just the leaves are almost drying. Then management of seedling blight. One option is seed dressing with 4 gram per kg seed Trichoderma viridi. So Trichoderma viridi is important, and then soil drenching with copper oxychloride at the rate of 3 gram per liter or metalaxyl 2 gram per liter. So drenching can be done, and remove and decimate tainted plant deposits. Next important disease of castor is Alternaria blight. So it is caused by the fungus Alternaria ricini. All aerial parts including stem, leaves, inflorescence, capsules may be attacked. Spores are irregular, scattered and have concentric rings. So concentric rings means round rings. These are brown and later become covered with bluish green or sooty growth. Sooty means black growth. In severe attack, spores coalesce, forming big patches and premature defoliation of plant, which gradually wilts. In one case, the capsule When half mature wilt suddenly turn brown and due to collapse of pedicel the capsules fall. They are similar in size and have underdeveloped and wrinkled seed with little oil contents. So it not only reduces the quantity of the seed, but it also reduces the quality of the seed. And you can see the symptoms on the right side, symptoms for Alternaria ricini or Alternaria blight. Seed treatment may be useful in combating the initial phase of the disease. Use healthy seed and treat the seed with Captan or Thiram. These are the fungicides at the rate of 4 gram per kg seed, or in a standing crop spraying of Mancozeb at the rate of 2.5 gram per liter at an interval of 15 days commencing from the 90 days of crop growth. So this is how we can chemically control the Alternaria blight. The next important disease of castor is Cercospora leaf spot. It is caused by the fungus Cercospora ricinella. So disease appears as minute black or brown points surrounded by a pale green ring. Spores visible on both leaf surfaces. So you can see these kind of black or brown spores on the right side on the leaf. As spores enlarge, they increase in size, center turns pale brown and then grayish white, surrounded by a deep brown band which may be narrow and sharp or broad and diffused. The fructifications of the fungus. Fructifications here means the fruiting bodies of the fungus appear as tiny here means small, black dots in the white center or in the reticulum. Diseased spores often occur in great numbers, scattered over leaf and roundish when young, but may become irregularly angular when mature. So they also change their shape. When spores are close together, intervening leaf tissue withers and large brown patches of dried leaf may result. Spraying with Mancozeb at 0.25% and the quantity of solution as usual will be 500 liters per hectare, and spraying twice with Mancozeb 2.5 gram per liter at 10 to 15 days interval reduces the disease incidence. And another option is treat the seed with Thiram or Captan 4 gram per kg seed. And in case of disease management also, crop rotations are important. Intercropping of the system is also important. Therefore, it is advisable to go for diversification of the crops to reduce the insect pest and of course the diseases. Now next important disease is wilt. It is caused by Fusarium oxysporum, F species ricini. So in this case, vein clearing in young leaves and yellowing and drooping of old leaves is followed by stunting of plant and marginal necrosis. Severe attack causes death of plant and infected seeds facilitate the spread of the disease. So it may come from the seed, and in wilt, as most of us understand, that this fungus grows in the vascular system of the plant, particularly in the xylums, and then it stops the translocation of water and other constituent in the plant system, and then plant may dry and die. So in this case, to control the wilt, selection of disease-free seed is required. So we can grow varieties which are tolerant to wilt. It includes varieties like Jyoti, Jwala, GCH4, DCH30. And seed treatment is also important because partly it is seed-borne disease. So seed treatment can be done with Thiram at the rate of 3 gram, or if you take carbendazim, it can be taken at 2 gram. And if you take Trichoderma viridi, it can be taken at 4 gram for treatment of 1 kilogram of seed. And next important disease of castor is powdery mildew. It is caused by the fungus Levilula torica. It is characterized by typical mildew growth, which is generally confined to under surface of the leaf. So in this case, powdery, a kind of powder looks to be spread on the surface of the leaf. In severe infection, the upper surface is also covered by the whitish growth of fungus. Light green patches corresponding to diseased areas on undersurface are visible on upper side, especially when leaves are held against the light. Let us see the control measures for powdery mildew. So when weather is dry, spray twice wettable sulfur 3 gram per liter at 15 days interval starting from 3 months after sowing, because it is partly a longer duration crop. So in this case, wettable sulfur can be applied. The rate of application is 3 gram per liter and it should be used. Then spray 1 ml of hexaconazole or 2 ml of dinocarp in 1 liter of water at fortnight interval. Fortnight interval here means at 15 days interval. Variety Jwala is resistant. So it is always advisable to go for integrated management of fungal diseases or any other diseases. In integrated management, we should follow crop rotations, we should go for deep summer plowing, and also we should go for cultural practices that can reduce the incidence of diseases. Now next is number 6 is root or charcoal rot. This is also important disease of castor and caused by the fungus Macrophomina phaseolina. Sudden wilting of plants in patches under high soil dampness stretch combined with high soil temperature is a typical side effect. Inside 7 days, leaves and petiole hang down, and inside a fortnight, the tainted plants go away. So in this case, the infection is very fast and the damage is also quick. So dim dark colored injuries are seen on the stem close to the ground level. Now seed treatment is an option of Trichoderma harzianum followed by soil application and drenching of same bioagents. So Trichoderma harzianum is excellent fungicide or kind of fungus actually, bioagent which control other fungus. So besides this, Trichoderma viridi is also used as a bioagent, and these kind of bioagent are finding their prominence, their use in agriculture these days, particularly in organic farming, because chemical fungicides are not allowed. So under those conditions, Trichoderma harzianum or Trichoderma viridi can be used. The usual rate of application for seed treatment with this bioagent is 4 gram per kg seed. So 4 gram per kg seed treatment can be done with Trichoderma harzianum to control the root or charcoal rot. Soil drenching with carbendazim at the rate of 0.1% 2 to 3 times at 15 days interval. So drenching here means you are just putting a lot of water along with fungicide, means excess amount of solution is put into the soil and it controls the fungus, or just washing of the soil surface. And drenching should be done 2 to 3 times at 15 days interval, and 0.1% solution of carbendazim should be made. It is also called as bavestine. So that means you can have 1 gram bavestine per liter of water to make the solution 0.1%. Next is grey rot or mold. It is Botrytis ricini. So ricini name comes from the castor, and most destructive disease of capsules. So capsules, as you know, are the fruiting bodies containing the seeds. So it can affect the capsules as well as seeds, and bluish spots on inflorescence and developing seeds. So these are the identification marks for this disease. Later circular or elliptical sunken dark colored spots appear. So one can notice the dark colored spots which are circular or elliptical in shape. Both flowers and young capsules become soft or whole capsule is destroyed. So many times you can find lot of damage to the flowers, to the fruits, and also to the seed by the grey rot or mold. Carbendazim as usual 50 WP at 0.1% is best treatment when sprayed after removing infected spikes. So, of course, this bavastine or carbendazim is quite popular fungicide. It is cheaper also, and rate of application is 0.1%, and which can be made by dissolving 1 gram of carbendazim or bavastine in 1 liter of water. Such kind 500 solution should be used per hectare basis. If disease continues, the same kind of spray can be repeated after 15 to 20 days. Now, next important disease is bacterial disease, which is called as bacterial leaf spot. It is caused by Xanthomonas ricinicola. It affects the cotyledons, leaves, veins, and creates few to various little round water-soaked spores, which later end up dark colored to coal black color. So, as you can see that this disease can come at any stage, particularly starting from germination, field emergence, vegetative stage, or even during the reproductive stage. So, one need to be very, very cautious and careful in identifying this disease. And one can identify it easily by bacterial ooze. Some kind of liquid will come out from the leaves, and it can be seen on both the sides of the leaf. Expert people can easily identify the bacterial ooze from the leaves, and it confirms the bacterial infection. So, for control of bacterial leaf spot, hot water treatment of seeds at 50 to 60 degrees centigrade for 10 minutes. So, this hot water treatment can be done with the help of vapors also. And spraying of antibacterial material like streptocycline at the rate of 500 ppm mixed with copper oxychloride 0.3%. So, you can make two solutions separately and combined solution of about 500 liter may be sprayed per hectare, and it is very easy to prepare 500 ppm of solution of streptocycline. To prepare 500 ppm solution, we need 500 milligram of streptocycline in 1 liter of water. Now, by now you have seen the control of insect pests and diseases. In all these cases, it is really recommended to go for integrated management of insect pests and diseases. In integrated management, we can follow some cultural practices, some mechanical practices, some biocontrol measures, and all kind of measures should be combined so that our environment is not affected, our soil health is not affected adversely, and also the income of the farmer increases. If we rely only on chemical control of insect pest diseases, it is not good for the environment. Soil can have lot of chemicals load, and finally they may reach the human food also. So, therefore, we should go for integrated management of insect pests and diseases. Now, we switch over to the next topic, which is harvest and post-harvest care. So, harvesting of spikes should be done at right time and early to avoid capsule dehiscence resulting in loss of productivity. So, right time of harvesting is very, very important, and in this case we can remember one English proverb, a stitch in time saves 9. If we harvest the crop at right time, we can harvest more seed and more profit. So, because there may be some dehiscence here means the split of fruit. The fruit will split open and seed shattering can be there. So, that way the seed losses occur if we do not harvest at right time. So, it is estimated that about 15 to 20% of the produce is lost in the field due to the dehiscence. Dehiscence of capsule means opening of capsules and losing the seed. Under rain-fed situation in Andhra Pradesh, Telangana and Karnataka, due to waiting for different order spikes to come to maturity for single harvesting. So, this is major problem. So, in this case, several flushes of maturity comes. So, we need to decide the right time for several flushes. So, in this case, it is advisable to go for 2-3 times picking of the capsules. Now, whereas in Gujarat, harvesting is done at the green stage of the capsule, and multiple harvests are made. So that is really a good idea. Multiple harvest means harvesting the matured fruits only. Odor and maturity of the spike result in higher yields and low physical loss due to capsule and seed dropping or shattering. So if you harvest slightly immature capsules, then shattering losses can altogether be avoided, and this is definitely a wise practice. When the capsules turn pale yellow and a few capsules dry up, the spike can be harvested and dried for threshing. So this is a good idea to understand the right time of maturity by seeing the color of the capsule. So when the color of the capsule turns pale yellow from green, then it is a right time to harvest the capsule or to pick up the capsules. Harvesting by cutting the entire spike. So spike, one spike may contain many capsules, and capsule may have seeds. So harvesting by cutting the entire spike from the plant is followed in Gujarat, and after sun drying, threshing is done to obtain seeds. So it is necessary to go for sun drying before threshing the seed. Sun drying removes the moisture and it saves the infestation of the crop by insect pests and diseases, and also during storage. In Andhra Pradesh, Telangana and Karnataka, the dried capsules on the spike are plucked out and collected and threshed. Let us see the yield potential of castor crop under different situations. On average, about 0.8 to 1 ton per hectare seed is obtained. 0.8 to 1 ton means 800 kilograms to 1000 kilograms of seed yield is obtained per hectare, and about 2 to 3.5 tons per hectare seed is obtained under irrigated conditions. So you can see here, under irrigated condition, yield potential is almost double or more than double. So it can be about 2000 kilogram to 3500 kilogram per hectare seed is obtained, depending on genotype and growing conditions. Of course, these are the average values, but exact yield will be determined by the climatic conditions, genotype, and also the management practices. But these are just average values. Yields as high as 6 to 8 tons per hectare, or you can say 6,000 to 8,000 kilograms per hectare have been obtained by farmers in Gujarat regularly under high input management. That is why you have seen that the Gujarat state has highest productivity of this crop. Gujarat also have highest area and highest production of this crop in the country. Stubbles and shelled capsules can be incorporated into the soil, which on decomposition can add organic matter. So whatever residues, crop residues or plant materials we get, we should plow it back into the soil so that we can compensate for the losses made by the plant uptake or soil erosion or by other means of nutrients. Better to recycle these, and after decomposition of such material, the soil will receive the humus and it will also receive some nutrients and increase microbial activity. So they can also be utilized as compost or vermicompost, which in turn can be applied to agricultural fields to improve soil fertility. So these kind of crop residues can be recycled by producing the compost or by producing the vermicompost ex situ, means one can collect all these residues and prepare the compost, and same compost can be applied to the soil, and this will have enormous effect on the seed yield as well as on the health of the soil. In case of castor, leaf fall is also there, and this leaf fall contributes both as surface mulch and as a source of nutrition. So leaf fall is there and it covers the surface of the soil, thereby it regulates the temperature of the soil, it moderates the temperature of the soil, and it also reduces the evaporation losses of water from the soil surface. Now you can see some toxic constituents are also there in the castor seeds. So castor seed contains an alkaloid named as resinine, which is mildly toxic. This resinine is mildly toxic, and a protein resin, which is extremely poisonous. All these toxins remain in cake and do not get extracted into the soil. So that is why we know that this castor oil is generally, or not generally, it is not used by humans. It is not fit for human consumption, mainly because of variety of alkaloids or variety of toxic materials in this. All these toxins remain in cake also and do not get extracted into the oil. So therefore, this kind of cake is also not fit for animal feed. The ricinin level in seed ranges from 87 to 150 milligram per 100 gram. So therefore, this can find some other uses. The ricin content of the cake is 8 milligram per kg. That means you can say it is 8 ppm, and allergen is 12 to 14%. Repeated intermittent steaming is a simple method of removal of toxin. So there are number of methods to remove this toxic material, particularly reducing the ricin content in the seeds or cake or in the oils. So one option is repeated intermittent steaming. It can reduce the content of the ricin in the cake. Ammoniation is another process which, apart from detoxifying, increases the nitrogen content of the cake. So this becomes very, very important because it is increasing the concentration of nitrogen in the cake. And this cake can be utilized to increase the soil fertility. Particularly wherever we have low fertile conditions, this cake can be used. So castor cake is good source of nitrogen, and it can also kill certain fungus or fungi which are living in soil. So it has multi-purposes, and it will supply nitrogen to the soil in a slow manner or slow fashion. And finally, it will also increase the soil structure if we continuously use this kind of cake material to the soil. Castor cake, it is an excellent source of organic fertilizer containing 4.6% nitrogen, 2.6% phosphoric acid. Here phosphoric acid means P2O5, and 1% potash. Potash is in form of K2O. Further, it decomposes gradually and releases nitrogen slowly. So it acts as a slow release fertilizer also. In addition, in dry land, it helps in retaining the soil moisture for longer periods and helps in overcoming short dry spells. So what happens, this castor cake upon decomposition produces humus material. If we apply it continuously, there would be accumulation of humic substances in the soil, and these humic substances will bind the soil particles together, and it will help in increased aggregation of the soil particle, and this will eventually results in the increase in water holding capacity of the soil. So overall, you can see that addition of this cake will increase the water holding capacity of the soil, besides supplying the nutrients at a slower rates. So in addition, in dry land, it helps in retaining the soil moisture for longer periods and helps in overcoming short dry spells. In Gujarat, castor cake is intensively used in paddy fields and also for the control of termites. Industrially, the soluble globulin obtained from cake contains 18% sodium glutamine, which acts as a good appetizer. Further, castor proteins are good source of synthetic and textile fibers. Using castor cake as a feed after detoxicating by heating. Components of I&M, of course, highest production of BT cotton with a higher soil fertility balance. And de-oiled castor cake is a valuable organic manure, as has been discussed, particularly for organic farming, with dual benefits of highest nutrient content and nematicidal properties. Dear students, I hope you enjoyed this lecture. Thank you very much.